Hi everyone, Dr. Zajac again. Welcome to Lecture 3 in the gastrointestinal or digestive system physiology component of the lecture series. In today's lecture, we're going to finish talking about the motor activity in the stomach. We've already talked about what goes on in the fasted state in the orad and caudad parts of the stomach. We talked about what went on in the orad part of the stomach in the interdigestive state. We've mentioned the MMC motor activity that goes on in the caudad stomach in the interdigestive period, but we haven't really looked at it in detail. So we'll start there, then we'll move into and talk about gastric secretions, about what goes on in terms of digestive function in the stomach and absorptive activity in the stomach, and then we'll move into and begin and get largely through our discussion of the exocrine pancreas. So that's where we're going in today's lecture. Let's go ahead and move on. Like I said in the previous lecture, we had discussed motility in the orad stomach in both the fasted and the fed state. And we talked about the motility in the fed state in detail, about the continuous cycles of propulsive and retropulsive motor activity against that highly contracted pyloric sphincter that led to the trituration, pulverization, and production of chyme, and then the coordinated activity that moved that chyme into from the stomach through the pyloric sphincter and into the small intestine. We mentioned that the caudad part of the stomach, when we were in the interdigestive between meal state, was undergoing MMC type peristalsis, but we didn't talk about that in detail. So that's what we're going to concentrate on here at the beginning of the lecture. The lower half of the stomach, the caudad part, and specifically its motor activity, this MMC, which stands for migrating motor complex or migrating myoelectric complex type contractile activity, and that's an intermittent type of peristalsis. So remember, in the fed state, we had that continuous retropulsion, propulsion, retropulsion as long as food was there, continuously until food left, but we're doing just intermittent peristaltic contractions in the interdigestive period. So this slide is actually looking at this sort of MMC motor activity. It's actually looking at it in the small intestine, but what I do want to drive home here is that it's both the caudad part of the stomach and the entire small intestine, the duodenum, the jejunum, and the ileum, that is engaging in this same type of motor activity the whole time. So you can translate what we're showing here going on in the small intestine to what would be going on up here in the caudad part of the stomach. What you have is this migrating myoelectric complex. This is all the motor activity that's shown here on the purple side of this slide. And what this is showing is that when you have this MMC motor activity going on in any one given part of the tract, you'll have a long period where there's very minimal electrical and associated motor activity that goes on at all. This is a really long period, is the longest period. It's called the quiescent phase. And then you'll have a period of increased electrical and motor activity, but the motor activity isn't very well coordinated and it's not highly propulsive. And then you'll have a period of very high intensity, very well coordinated, very peristaltic contractions that are really propelling stuff long distances through that segment of the tract. And then very rapidly you fall down and you resume this quiescent, relaxed period for long periods of time. And then the process will repeat in that same spot again, long period of rest, repeat again and again and again. And it will keep doing this activity over and over again until you eat. And when you actually consume a meal, then it will kick you out of this MMC migrating myoelectric activity. And in the stomach, you would go into that continuous peristalsis that we've talked about. In the small intestine, we'll see that it will kick you out of this and move you into segmenting type contractions. And we'll talk about those when we talk about the intestine. So with this MMC, migrating myoelectric complex activity, It is a pattern of activity. It's one of those pre-programmed patterns of activity that's coordinated by the enteric neural networks in the enteric nervous system, in the stomach and the small intestine in the fasted state. It has multiple phases. So in phase 1, this is that long period of quiescence when there's that minimal motor activity and electrical activity going on. It's the longest part of the phase, and it will last for about 90 to 120 minutes. And then you'll get phase 2, and in phase 2 you have some rising electrical activity, and it stimulates a little bit of motor activity, but it's weak and it's not well coordinated and it's not highly propulsive. So that's phase 2. And then you get into the phase 3, and this is this part of the motor activity, and it's very, very, very high intensity, high amplitude contractions that are very well coordinated and highly propulsive. And so if you remember when we talked about the stomach in the fed state, we said that pyloric sphincter was neck down and it was really tight and it was having this really big seething activity. But we said that when you're in the interdigestive period, which is what you would be in this period, that pyloric sphincter tone is much more relaxed. It's still forming somewhat of a barrier to flow, but it's not neck down near as tightly. And so it's not making as much of a barrier to flow as it does in the fed state. And so when you get these powerful phase 3 contractions, what they're meant to do is if you had any particulates that were in here that during the fed state you couldn't pulverize, triturate, and get down to that 2 millimeter or smaller size limitation that you needed to be able to get through that constricted pyloric sphincter, now because you have these super high powerful contractions and a more relaxed pyloric sphincter, you could use those phase 3 MMC contractions to sweep out and empty out the contents of the stomach that you couldn't get pushed out before. So I'm sure you've all heard tales, or maybe yourself have witnessed, maybe you yourself did it at some point, swallowed some coins, you got rushed to the doctor or the emergency room, they're like, oh my God, they swallowed a penny, what are we going to do? And once the healthcare providers figure out that the swallowed particulate didn't go down into your airways, it's not clogging your airways, preventing your ability to breathe or anything like that, what they'll usually say is just go home and wait for a couple days and check the stools, and eventually whatever it is will work its way out. And so that's what these housekeeping peristaltic contractions are meant to do. They can clear the pipes and clean out that stuff in the stomach or in the small intestine. Remember, your small intestine ends at the ileocecal junction, and beyond that is the cecum and all that bacteria that's down in your large intestine. And some of that bacteria is also always migrating upward through the small intestine. So these MMC contractions, many of them start up here in the stomach. They sweep through the caudate end of the stomach and then through this junction and all the way down to and through the whole small intestine. So there are these housekeeping waves that just periodically sweep through, and anything that didn't clear it out, boom, they sweep it right down to that ileocecal junction. And if that sphincter happens to open up, they might sweep a little bit of it into the cecum, and the rest of it just gets retained down there. But it's doing these housekeeping sort of contractions to keep everything cleared out in between meals. Some textbooks will also discuss a phase 4, this little rapid period where you have a rapid decline of electrical and motor activity that then quickly brings you back to the baseline quiescence again. Many textbooks will just consider that part of the beginning of the quiescent period. The big thing is if you look at any given place in the tract, like we said, these waves are going to go through that same region again and again and again, with a periodicity of occurring about every hour and a half to 2 hours. But if you watch how this motor activity moves along the length of a gut, you'll see that one given wave will start here. There will be a little lag period, and then you'll get a similar contractile ring that starts a little bit further down in the system, and then there's a lag period, and then there's another wave that will start a little bit farther down. And the net effect is that this wave propagates the entire length of the caudate part of the stomach and the small intestine as each wave progresses through. Now I said the nervous system, ENS reflex arcs are coordinating all this neural activity and stuff, but this is a pattern of motor activity that is very largely dependent upon the permissive effects of a hormone called motilin. So motilin comes from a population of epithelial cells down in the small intestine called Mo cells, and they secrete this hormone with a cyclic nature. So its levels ebb and flow. They increase, they decrease, they increase, they decrease. And the levels of the hormone increase, and they reach their maximum levels right before the M3 motor activity. So as they're increasing, they will eventually get you, they'll get high enough that they'll let some of this motor activity actually result in contractions, but those contractions aren't highly organized. When these levels reach peak levels, they're able to really highly coordinate those motor activities and prime the tissues effectively so that these can be very powerful, very well coordinated propulsive contractions. And so if you block the secretion of motilin, or if you give neutralizing antibodies that block its effect, block the receptors on which it acts, then you actually do away with this MM3 type phase 3 motor activity. So it's very important in being able to get these effective peristaltic contractions coming through in this cyclic fashion. And again, just to emphasize, the MMC motor activity in the fasted state is giving you housekeeping functions that are just pushing all those acids down and keeping all that bacterial load down, way down at the distal end of the small intestine, ready for movement into the large intestine when the next meal comes along. What we want to move into and talk about today is the stomach. What goes on in the stomach? Well, we know that one of the main functions of the stomach is to provide that storage, reservoir capacity, let everything roll around in the acids that we've preformed before foods even got there, and that's for that protective immune protection benefit. Minimize the bacterial load you're going to deliver further on into the small intestine. How about digestion? What goes on? Well, the digestion that we started in the oral cavity, and you remember that was the digestion specifically of carbohydrates, and even more specifically of only the starch and glycogen type complex polysaccharide type of carbohydrates. So that's going to continue for a while, but the alpha amylase that's doing that enzyme activity is very susceptible to denaturation by all the acids present in the stomach. So as soon as that amylase comes in contact with the gastric acids, it's going to be inactivated. At the point at which all of it has been completely solubilized and mixed with gastric acids and inactivated, then you're going to temporarily halt carbohydrate digestion in the stomach, because the stomach does not secrete and add any new and additional enzymes that could do any additional carbohydrate digestion. What about lipid digestion? If we did have lingual lipases up in the oral cavity, we said before, they weren't going to be maximally active because the lingual lipases are an acidic lipase, and they need a very acidic pH to have any kind of significant activity, and you don't typically have that type of a pH environment in the oral cavity. When you swallow saliva down into the stomach, in species where there is lingual lipase there, it gets even more active than it was in the oral cavity, because now it's at its right pH environment. In humans, we don't have a bunch of it, so it's not a big deal for us. But what does happen in the stomach is the stomach is going to actually give us gastric lipases. So put some new lipid digesting enzymes in. These are going to come from chief cells primarily in the fundus region of the stomach, and they're going to start working on our triglycerides. We'll talk about that in more detail later. Protein digestion. Put a big star by protein digestion. This is the first place where you have the potential to have protein digesting enzymes. And we're going to find out that in the stomach we have chief cells, and they're going to secrete a precursor called pepsinogen that is inactive in the form it's originally made, but it can undergo a cleavage reaction and form pepsins. And those will be enzymes that actually do start to chemically modify proteins in the stomach. We'll talk about that in more detail later. What about absorption that goes on? Turns out that all of these acids that are working in the stomach and all these digestive enzymes that are going to be there aren't going to produce a whole lot of absorbable end products. There is going to be some minimal absorption that can go on across the wall of the stomach, but mostly it's going to be very small molecules that can get through the gelatinous water layer, that mucousy water layer that's hydrating and protecting the epithelium there, and then still are small enough that they can readily penetrate the lipid bilayer and be absorbed into the bloodstream. So the things that can be absorbed across the wall of the stomach are going to be small amounts of alcohol, small amounts of caffeine, very short chain fatty acids, and small organic acids like aspirin, like sulfosalicylic acid, which is what constitutes aspirin. So a few pharmacological agents, things like that. Those are the main things that are going to get across the wall of the stomach, and everything else is going to be passed on into the small intestine, need further modification down there to make it into absorbable end products. So we've talked about how the motility functions are really important in the stomach in forming chyme and then regulating the delivery of chyme into the small intestine. I just refer you to go back and review those notes on motility from the last lecture. But we also know that in addition to motor functions, which subserve digestion and absorption, there's also a lot of secretory functions that are going to subserve these processes too. So what kind of secretory products are going to be secreted from the stomach that will be important in gastric acid function or in gastric function? The number one thing is acid, and specifically we're talking about hydrochloric acid, HCl. This is the main acid that comes from the stomach epithelium. It's going to be produced by parietal cells. So that's a main product. We're going to talk about how it's made, where it's made, how it's regulated. Gastrin, histamine, and somatostatin are all substances that are also going to be secreted in the stomach, and they are major regulators of acid production. So gastrin is going to be a stimulant. It's a hormone that will act in true endocrine fashion to turn up acid secretion. Histamine is going to be an amino acid signaling molecule that's going to act in paracrine fashion also to turn up acid secretion in the stomach, and somatostatin is going to be a paracrine agent that can act to turn down acid secretion and histamine secretion and gastrin secretion. Somatostatin likes to turn everybody off, so it's going to be a regulator that turns off all of those things, and collectively all of these agents regulate acid secretion. We're going to talk about pepsinogen. This is that inactive precursor that can form pepsin and lead to the initiation of the protein digestion that's there. This is going to come from chief cells in the stomach. We'll explore that in detail. We're going to talk about intrinsic factor. This is a second factor that comes from the same cells that make the acid, also comes from the parietal cells. And intrinsic factor is going to be very important in helping us to absorb vitamin B12. We'll talk about that in detail in today's lecture. Lastly, the stomach is making all of these digestive enzymes and the acids, and those could chew the stomach up itself, so it has to have a number of protective mechanisms to prevent its own autodigestion, and chief among those are the secretion of mucus and bicarbonate molecules that are very important in maintaining the integrity of the epithelium in the stomach lining, and also prostaglandins, PGE2 type prostaglandins. So we'll talk about those in today's lecture. Before we really get into and begin discussing those things in detail, we should talk about some important modifications that are present in the gastric epithelium that are really essential and fundamental to the stomach being able to do what it does. So first of all, you'll remember when we talked about kind of the tissue paradigm, the tissue layer paradigm of the GI tract, we talked about that muscularis externa, that big thick muscle layer whose motor activity drove all the tonic contractions in the sphincters and all of the phasic, segmenting, and peristaltic contraction that move things through in regulated fashion through the tube. And we said that in most places that muscularis externa was a two-layered muscularis externa. So internally, close to the submucosa, you had an inner circular layer where the muscle fibers were oriented perpendicular to the long axis, and when they contracted, they narrowed the lumen of the organ in that area. And then deep to those deeper in the wall between the circular layer and the connective tissue protective layer outside, you had the longitudinal, outer longitudinal muscle layer, and those muscle fibers ran parallel to the long axis. And when they contracted, they would shorten the length of the tube in this particular area. The stomach is a unique part of the whole GI tract because it has a 3-layer muscularis externus. So it has an additional layer that we call the oblique layer. In the stomach, the longitudinal layer is still the outer layer just under the submucosa. And the circular layer lays right next to it, just inside it. So where you have this third oblique layer is inside, underneath the circular layer. So the oblique layer has its fibers oriented diagonal to the long axis of the gut. So it can actually drive contractions in a third plane because these are diagonal to the long axis of the gut. And this muscle layer lies then between the circular layer and the submucosa that's inside of it and closer to the lumen. So it's a 3-layered muscularis externa, and the third layer, like I said, lets it contract in 3 planes instead of 2, and that's very important in letting the stomach do all of the triturating and the grinding and pulverizing and stuff that it needs to do to create chyme. Then if we opened up the stomach and we looked at the interior lining, the epithelial lining inside underneath the muscle layer, we see that the inside is folded into these very big up and down longitudinal folds. And these longitudinal folds form what we call the rugae or rugae. And the function of these is that we know that when we're eating a meal and we have that receptive relaxation going on and the accommodation, so we're relaxing all the muscle in the wall out here, we also have these pleats in here that as we start moving in a lot of meal constituents into the stomach, these pleats can simply unfold and widen out and expand within that relaxed gastric wall and make more room for those nutrients to come in. And that's very important in allowing us to be able to consume a high volume of nutrients without having a huge rise in intragastric pressure. That would make us feel uncomfortable, have a stomach ache, not want to take in any more nutrients or anything. So that's really important. Then if we look at the stomach surface itself, the epithelial surface lining, we see that it has lots of these little pits within it. And what are these pits? These pits are areas where a number of invaginating ducts, you'll have, or glands, excuse me, invaginating glands, about 4 or 5 different glands will come up and they will meet just underneath the surface of the epithelium and empty into one of these little pit areas. And so the pits are the sites where we gain access to and where things exit from these gastric glands. So if you remember when we talked about the stomach in the last lecture and we said you could divide it if you were talking about its motor functions into 2 main regions, the orad and the caudad parts of the stomach. And those are regions that differ in terms of their motor activity, their motor function, their contractile activity. But you can also differentiate the stomach and divide it into a number of regions based on the digestive processes that it does, the different types of cells that are there, the different types of secretory activities and products that it makes, secretory activities it has, products that it makes, and the different contributions the different regions make to the digestive process. So if we look at it in that way, we can divide it into broadly 4 main areas. This area right around where the esophagus and the stomach join, this is what we call the esophagogastric junction. The area that encircles this region right here is called the cardia region. The area where we said it's a big J-shaped organ, and the area where it necks down and gets really narrow down here at its very distal end by the pyloric sphincter, that we call the antrum. The area lying just to the left of and extending above that gastric esophageal juncture is what we call the fundus. And between the fundus and the beginning of the antrum, we have the body or the corpus part of the stomach. Some textbooks that you read may actually include the fundus as part of the body. They just say specifically that it's this region that projects to the left of and rises above this gastroesophageal juncture. So those are the main regions that differ from one another functionally because of the types of cells they have, the types of secretions they make, and the contributions that they have to digestive and absorptive processes. So when we look at these glands, it's important to note that we have two different types of glands. Up in the body region of the stomach, we have oxyntic glands. And to be considered an oxyntic gland, you have to have a particular type of cell called parietal cells. And these are the cells that are responsible for the secretion of the gastric acid, the hydrochloric acid, and the intrinsic factor secretions that come from the stomach. So the glands that are in this region have among their constituent cell types parietal cells. The glands that are down in the antral region lack those acid secreting cells, and they are mostly mucus secreting glands, and they will have a preponderance of endocrine cells like G cells and D cells, but they will be non-acid secreting. So the antrum lacks parietal cells, and the cardia up here actually also lacks the parietal cells. All of the stomach will have, many of the glands here will also have chief cells. These are the cells that make the pepsinogens, and the chief cell population that is up here in the fundus region of the body is where those those gastric lipases will come from. So there are all of these different cell types in the gastric epithelium, and we'll talk about them when we're talking about all these other functions throughout the rest of the lecture. So it's important to understand what these cell types are and what are the major secretions that they provide, or the functions that they provide in the stomach. So the ones that are up here on top and in direct contact with the lumen are what we call SEC cells, or surface epithelial cells. And they secrete a lot of fluid that is rich in bicarb and that hydrates and provides bicarb on the luminal surface of those cells. And then you have, coming down into the gland, you have mucous neck cells. Mucous neck cells, along with chief cells, can be a source of pepsinogens, and they make a lot of mucus. And mucus has, again, those mucin glycoprotein molecules in it, like we saw in the salivary glands, and it provides that lubricating, protective, gelatinous coating that is protective to the cells. Because the stomach makes all of these acids and all of these digestive enzymes that could be very injurious to the stomach itself, lots of the cells there, it creates a very hostile environment. And the cells are getting insulted by these enzymes and the acids all the time. So they become rapidly damaged and dysfunctional, and they will be shed off. And to maintain the integrity of the gastric epithelium, they need to be replaced by other cells. So importantly, in the glands, in kind of the mid-region glands of the stomach, we have stem progenitor cells. So these are undifferentiated pluripotent cells that have the capacity to produce cells that will differentiate, can migrate upward, differentiate, and become neck cells, mucous neck cells, or surface epithelial cells, or can migrate downward and become acid intrinsic factor secreting parietal cells, pepsinogen secreting chief cells, or lipase secreting chief cells, or even make some of the endocrine cells that are down in the very base of the gland. And these would be cells like the G cells and the D cells. So going back to the parietal cells, those are the cells that are before I go there, I guess I should remind myself here to say that because this environment is so hostile, these progenitor stem cells can turn over and completely repopulate the whole lining of the stomach down deep through the glands and all the surface epithelial layers and stuff every 2 to 3 days. So this is one of the most highest cell turnover rates seen anywhere in the body. And that's because it is that this environment is so injurious to the cells. Because these cells have developed into cells that have such a high turnover rate, they are also susceptible to the injurious effects of cancer therapeutic drugs that are designed to target and remove rapidly dividing cells like cancer cells. A negative consequence of those type of drugs is that they can't differentiate between what's a rapidly dividing cancer cell and what's a normally rapidly dividing cell in the stomach. And so they will oftentimes cause injurious effects on the gastric epithelium, will cause a lot of death and destruction of those cells. And this is what makes people who are on chemotherapeutic regimens have lots of trouble with nausea and an inability to eat, lack of appetite, and all that sort of stuff when they're going through those treatments on certain sorts of chemotherapeutic regimens. The parietal cells are the cells that make the hydrochloric acid itself and the intrinsic factor important in B12 absorption. Like we said before, they're not present in the glands in the antrum and down in the pyloric region. Chief cells are the cells that make the pepsinogens, and they make the gastric lipases up in the fundus. And then the endocrine cells, G cells, secrete the hormone gastrin. D cells secrete the paracrine agent somatostatin. ECL cells secrete the histamine. All of these agents, the D cells and the G cells, are in the bottom of the gland itself, in the glands down in the antral region of the stomach. And the histamine secreting ECL cells are lying in the lamina propria just underneath the surface of the epithelial cell layer, and they are releasing histamine that is going to act in paracrine fashion to influence the parietal cells and acid secretion. Let's begin our discussion of the secretory activities that go on in the stomach that subserve the digestion that's going to go on there by talking about gastric acid secretion. We know that the acid secreting cells in the stomach are the parietal cells. And the thing that enables these cells to actually secrete all of the acid that they produce is specifically the presence of these hydrogen potassium ATPase pumps that are on their apical membranes. These are going to spend cellular energy to transport hydrogen ions out of the cell against the hydrogen concentration gradient. They can build up hydrogen ion concentrations in the lumen that are 1,000,000-fold or greater higher than what you see in the interstitial fluids and the blood in close proximity to their basolateral sides. The cells have a unique structure. They're a very wide-based cell, so that there is a very wide basolateral side of the cell, and this cell has a lot of the transporters, a lot of the receptors that are going to pick up and bind regulatory proteins that are important in regulating parietal cell acid secretion. They also have these long invaginations along their apical surface that form what we call canaliculi. Each one is a canaliculus, or collectively they would be canaliculi. And these big, deep channels that invaginate deep into the body of the cell are continuous out here with the lumen of the stomach. They also have structures called tuberovesicles. In their resting state, these tuberovesicles are little membrane-bound vesicles that lie just beneath this canalicular apical membrane, and they are made with many of these preformed hydrogen potassium ATPase pumps embedded in their organelle membranes, and they have a lot of carbonic anhydrase enzyme present in those membranes as well. When the cell becomes active and it is stimulated, the activation causes a translocation of those preformed vesicles that contain all these hydrogen potassium ATPase pumps, causes them to translocate, move to the apical surface, and fuse with the apical membrane along the canalicular linings. This significantly increases the surface area projecting out into these canalicular channels and gives you a lot more surface area over which you now have expressed these hydrogen potassium ATPase pumps that are active in the actual hydrogen ion secretion itself. So what is going to actually activate these cells and get them going. There are three main secretagogues that will activate those parietal cells, and a secretagogue is just simply some substance that will stimulate the production of whatever it is you're talking about. So the secretagogues that are going to turn on hydrochloric acid secretion are primarily going to be acetylcholine. Acetylcholine is going to come from vagal nerve fibers. Essentially, you're going to have wall stretch, and that wall stretch is going to activate vagal pathways, and the long preganglionic fibers are going to come out. They're going to synapse with ENS fibers, and ENS fibers are going to be cholinergic fibers that release acetylcholine. The acetylcholine that's going to be released is going to bind its own type of receptors, which are going to be M3 receptors that are present on the surface of the parietal cells. Gastrin is going to come from the G cells that were down in the antrum, and gastrin, when it's released by those G cells, is going to be released into the bloodstream, and it's going to act in true endocrine fashion. It's going to be carried by the bloodstream and reach the basolateral cell surface through the bloodstream and the interstitial fluids. It's going to bind its own receptor, and it is a CCK2, or in older literature you might find this called a CCKB type receptor. The gastrin molecule itself is going to be very similar structurally, have a fair amount of structural homology with a hormone called CCK, cholecystokinin, that comes from eye cells in the small intestine. And both gastrin and CCK are going to, because of their structural similarity, conveniently bind very structurally similar receptor molecules. And the CCK1 receptor, or CCKA receptor, has a much higher affinity for CCK than gastrin. So CCK1 or CCKA receptors are going to mediate CCK effects, and the related but slightly different CCKB or CCK2 type receptors have equal affinities for gastrin and CCK, but gastrin is typically, during the course of a normal meal, released in much higher quantities than is the CCK, and so the CCK2 or CCKB type receptors are going to mediate primarily the gastrin response. And then lastly, histamine is going to come from cells that are the ECL cells, from enterochromaffin-like cells that are not in the glands themselves, but are located in the lamina propria support tissue that lies just beneath and supports and anchors the parietal cells in place. So the histamine secreting cells will be out here. They will release histamine, which is a water-soluble signaling molecule that's made from modifying histidine type amino acids. They will release that, and in paracrine fashion, it will diffuse over and bind to its own receptors, which are called H2 receptors. And all of those receptor subtypes are present on the basolateral sides of these cells. So all of these agonists can induce direct effects by being released, binding to their own receptors, and modifying acid output. And these same secretagogues will also be the secretagogues that will stimulate these cells to release intrinsic factor, although the intrinsic factor will be pre-made and stored in vesicles and released in by the activation of different intracellular proteins and trafficking proteins and stuff like that, but still be regulated by these same sort of secretagogues. There are you can look at the intracellular signaling pathways by which these substances act. The gastrin and the acetylcholine both bind G-alpha-Q trimeric protein coupled receptors that target and activate phospholipase C, convert PIP2 into diacylglycerol and IP3, so release increased amounts of calcium into the cytoplasmic spaces from the ER via the IP3 and activate protein kinase C through the diacylglycerol. The histamine, on the other hand, activates a different type of cell signaling cascade. It targets adenylyl cyclase through G-alpha S trimeric protein coupled receptors, increases the amount and activity of, or the activity catalytic ability of adenylyl cyclase, activates protein kinase A, changes a lot of phosphorylation states of proteins involved in the trafficking and the release of all of these ions essential to acid production. So because the gastrin and the acetylcholine set up and activate one type of intracellular signaling cascade, and the histamine activates a different cascade within the cell, these two can have synergistic effects and potentiated effects. If they are all present together, can give you much more of a response than any one of them present all by itself. There are also a number of substances that act to inhibit the acid secretion. So things that would inhibit it would be the somatostatin. Remember, this is going to come from the population of cells called D cells in the stomach lining. And we have PGE type prostaglandins, and PGE type prostaglandins and somatostatins both target G alpha I trimeric coupled protein receptors, and those would inhibit adenylyl cyclase, turn down pKa activation, turn down the phosphorylation that pKa induces, and those changes would have negative effects on all these transport processes that will be important to acid secretion. So that those would turn down acid secretion because of that. And then enterogastrones are a number of other substances. The term enterogastrone means entero, having to do with the intestine. So substances that are made in the intestine, gastrone, that happen to exert their influences down in the stomach. So enterogastrones will be things like CCK, cholecystokinin, or secretin, or gastric inhibitory peptide, any number of substances made down in the intestines that come back and act in the stomach to turn down acids or to turn down or to slow gastric gastroduodenal emptying and the rate at which chyme is delivered to the small intestine. That's the technical definition of an enterogastrone. Let's look at these cells in a little bit greater detail and see how they do what they do. First of all, we know that they're going to secrete all of this hydrogen and create a hydrogen gradient that can be a millionfold different in hydrogen content in the lumen of the stomach versus the interstitial fluid and the blood. So any cells that influence pH generally almost always express inside the cell carbonic anhydrase. So this is an enzyme that when water is present inside the cell and the water ionizes into hydrogen and hydroxyl ions, carbonic anhydrase can combine those hydroxyl ions with carbon dioxide and create bicarbonate. And it also, the ionization of that in these cells is going to be particularly important because it's going to provide some of the hydrogen that's going to be transported by those critical hydrogen potassium ATPase pumps. So those hydrogen potassium ATPase pumps on the luminal face, Their ATPase activity is important because it can spend cellular energy. It has the capacity, enzyme capacity to hydrolyze this, make energy available to drive the export of hydrogen against its concentration gradient. On the basolateral side of the cell, we have these sodium-potassium ATPase pumps. Again, their ATPase activity lets them move these two types of ions, each of them against their concentration gradients. So remove sodium from the cell and bring sodium or bring potassium into the cell. Make a big note here wherever you have these, and they're going to be on the basolateral sides of almost all the cells we'll talk about through the GI series. One of the big jobs that they do is keep the concentration of intracellular sodium low. And then many cells will use that low intracellular sodium gradient to move a lot of other things secondary to that sodium gradient. And so this is going to be true in these cells as well. We create a low intracellular sodium gradient through the actions of this pump, and that's going to power the transport of lots of other things important to this gastric acid secretion. So on the basolateral side of the cell, we're also going to have sodium hydrogen exchangers right here, and they're going to bring sodium in driven by that gradient that's established by these pumps in exchange for hydrogen that they take out. We're also going to have sodium potassium 2-chloride or NKCC transporters. They are going to bring sodium in according to its concentration gradient established by these pumps, but conveniently it's going to load up with potassiums and chlorides. So this is a way we're going to get a bunch of chloride ions into the cell. And this is bringing the chloride ions into the cell and the potassium ions into the cell. Against their concentration gradients. They're just coming with the sodium that's following its gradient. On the basolateral side of the cell, there's going to be potassium leak channels. So a lot of the potassium that comes in this way is going to leak out. A lot of the potassium that comes in here is going to leak out. But we're going to accumulate a bunch of chloride ions in here, right? So we have these types of transporters on the basolateral side of the cell that are important for getting again, some hydrogen in the cells that can supply hydrogen to these pumps, and also bringing chloride ions into the cell. We also down here have bicarb chloride exchangers. We'll see chloride bicarb exchangers throughout the GI tract. They're very, very common, and their presence on these cells on the basolateral side is going to bring in chloride ions in exchange for bicarb ions. And so a lot of the carbonic anhydrase-derived bicarbonate is going to exit the cells in this way and bring chloride in. So we have multiple mechanisms on this side of the cell to bring in chloride ions. We're going to spend all this energy to dump a bunch of hydrogen ions out here against their concentration gradient. So every time we pump hydrogen ions out here, that pulls this whole reaction and this reaction in the forward direction and dumps more bicarb out here. So these chloride bicarb exchangers, as we're dumping acid into the lumen of the stomach, are going to be really important in dumping a bunch of bicarbonate out into the interstitial fluids. And that creates what we're going to call an alkaline tide, because much of that bicarb will enter the blood. And so the blood leaving the stomach, when it's actively secreting acids, is going to have this alkaline tide that is carrying a lot of bicarb away from the stomach during these periods of time. So that's very important there, but it's also another mechanism that we can use to get chloride into the cell. So as this pump is very, very active and it's pumping lots of hydrogen ion out here, as the lumen becomes more and more electropositive because of these cations being delivered, the protons being delivered, that creates an electrical gradient that wants to pull anions in its direction. And the main anions that are going to come are going to be these accumulated chloride ions that have come in through these various mechanisms on the basolateral side. So there are going to be a number of chloride channels in the apical membrane. One of the big ones that we'll see again over and over in many places throughout the GI tract is a protein called the CFTR. This stands for cystic fibrosis transductance conductor regulatory protein. That's a big fancy name for just saying I'm a chloride channel. And when this channel is open, chloride can move through it bidirectionally. It will move according to its electrochemical gradient. Here, both the electrical gradient and the concentration gradients favor the chloride coming out into the lumen and following the hydrogen. And so that's how you get this HCl acid that accumulates. As all these ions accumulate out here, they're adding more and more and more solutes to the lumen out here, and that creates an osmotic gradient to pull water. So there are aquaporin channels in these cells that can allow water to come in and water to move out across both the apical and the basolateral sides of the cells. And generally we can have movement of, in many places, we would also see movement of water through these paracellular pathways. But here the water follows the acid out, and you get all of your acid secretion in this mechanism. So they're very critical to being able to move the chloride ions across the apical membrane. And another thing to remember is when you have these countertransporters, anytime you have a co-transporter or a counter-transporter transport protein, it has to load up at all its appropriate sites before it can work. And so that's why most cells that have the sodium-potassium ATPase pumps on their basolateral sides have potassium leak channels, so that at least enough potassium can leak out to reload out here to keep these pumps able to load at both locations. Here we're also going to have some critical potassium leak channels on the apical side, so that we can again get some of the potassium that's brought in through the actions of the sodium potassium pump to leak out here, and make sure that we have enough potassium out here to keep this pump able to load at both sites so it can keep operating efficiently. So I like to use this slide as a way of just kind of tying everything together about acid production. We know that the parietal cells make the acid. We know that these are the three secretagogues that turn them on and get them to do that. We know that the G cells make the gastrin. The ECL cells make the histamine. We know that vagal parasympathetic signals come in, and cholinergic fibers that release acetylcholine from postganglionic ENS fibers will turn these parietal cells on. What turns on the G cells? If there is simple stretch of the gastric wall, that stimulates them. They have sensors that can detect what's the luminal contents look like. If they detect the presence of amino acids there, they know that, for instance, they're especially sensitive to detecting aromatic amino acids like phenylalanine and tryptophan and tyrosine. And if they see those there, that turns them on. And lastly, there are a bunch of nerve fibers that project. Remember, these cells are down in the antral region of the stomach. So there are a lot of nerve fibers that project to them that synapse with and use what we call peptidergic ENS fibers as their postganglionic fibers. These release peptides, in this case gastrin-releasing peptide, which is a stimulatory protein that binds its own receptor and turns these cells on. So even before you put food into the stomach, when you're in the cephalic phase of gastric acid secretion, all your stimuli are coming from the head. You're seeing food, smelling food, tasting food, hearing food being made, or somebody eating food. You're looking at food in front of you on the TV, in a magazine, whatever. You might even have food in the mouth and you're tasting it. But the important point is you don't have anything in the stomach yet. But already all of these other sensory organs are collecting all this information that implies there is a likelihood that you might get some food delivered to the stomach. So they send this sensory information in to the center in the central nervous system that regulates efferent outflow of parasympathetic activity out to the organs of the GI tract, and they increase parasympathetic output. So you have all these inputs that are turning on the parietal cells, that are turning on the ECL cells, and that through the use of those peptidergic fibers are turning on the G cells. So you're turning on all the cells that would upregulate, kind of put the gas on, give the green light to that acid production before there's even food in the system. There's also a mechanism for braking or putting the brakes on acid production. So we have this other critical population of cells that are the D cells. And D cells in the stomach are considered the brakes for acid production because they're the cells that make all that somatostatin. And so when they make somatostatin, somatostatin acts in paracrine fashion over here to tell D cells, turn off your gastrin. That turns off direct effects up here, and it turns out indirect effects through making more histamine. And somatostatin that's made by D cells that are up in the body of the stomach, the same somatostatin comes and it binds its receptors over here and it turns these cells off. So that could put the brakes on acid production. Well, if you're in the beginning of the cephalic phase of acid production, you want acid up and running, and you want to be riding the gas pedal all the time and, you know, kind of guns out to give you acid production, and you don't want to be riding the brake at the same time. So very conveniently, these brake cells these D cells actually express a different type of receptor. And because they express different types of receptors, not the M3 receptors that are characteristic on most other cells, these receptors allow them to turn on different intracellular signaling pathways, and this actually has the effect of turning turning off their somatostatin secretion, right? But this brake can be turned on. It turns out that the D cells, especially those down in the antrum next to the G cells, they have the ability to sense the presence of hydrogen ion itself. So when the pH is very, very low, which means that the hydrogen ion content is very, very high, that can be a stimulus for turning these cells on. So these cells are really getting some signals that would tell them to turn off acid secretion, and they could be getting cells that would also tell them, that could be telling them to turn off somatostatin secretion. They could also be getting stimulatory cells to tell them to turn it on. What happens at the beginning of a meal is you actually have suppression of this somatostatin because the overriding input to these cells is coming from the vagal stimulation, and it's telling them don't turn on, don't turn on, don't turn on. So you're in that cephalic phase, and then you start to build up all of this acid secretion, anticipating foods are going to come down. If no food comes down, there's nothing in the stomach to neutralize that acid, and very quickly the pH drops to below 3.0, and that now becomes the overriding signal, and that will turn these cells on and tell them go ahead and release your somatostatin. So it turns down the G cell activity, turns down ECL histamine release, turns down direct effects up here, and it can actually then start to turn the cells down because the cephalic phase said we think you're going to get something, but you didn't get anything, and so now it has a way of turning its own self off. So this is a powerful negative feedback system whereby the acid that's produced and doesn't have anything to neutralize it can initiate a system of turning its own secretion off through activating this somatostatin-induced paracrine inhibition of G cells. Now, when else would this happen? When you get into the gastric phase of the secretory process, you've got food in, you're stretching the stomach wall, you actually do have luminal contents in the stomach now that can be detected, and so you turn on all of these stimuli, and you still have this vagal nerve input coming in and releasing GRP to tell you to turn these cells on too. So when food comes into the stomach, it will neutralize the brake, and it will neutralize, all the acid that's being produced. That will bring the pH above 3, and so it will temporarily turn the brake off. So you'll get even more heightened production of acid when you're in the gastric phase. But then the stomach is going to start delivering all of its chyme into the small intestine, and if you're not bringing any more food in, eventually, as you eliminate all the nutrients out of the stomach into the small intestine, all these things that are geared up and causing acid production to go on now aren't countered by anything that is neutralizing the acid that's being produced. So that will again start to drop the pH, and it will institute the brake action again, activate the D cells, cause the release of somatostatin, and turn the G cells off. And so this gives you maximum production of acid when you need it, and there are nutrients moving through the system, but this very effective negative feedback system will turn the acid production off when you don't need it. So that's critical. The chief cells, remember, are the cells that are going to produce pepsinogen, and one of the reasons acid production is so important is because the acid that you make will drive the very rapid conversion of this inactive precursor to its active enzyme, and the low pH environment in the stomach will give you the pH that's optimum for the pepsin, for the lipase that's going to be there, and will help facilitate digestion. So let's just summarize then what we've already said. Why is it important to make all this gastric acid? We know about the sterilizing effects that it has. We've talked about that multiple times. I didn't mention it, but it also denatures many proteins. Many of your ingested proteins get denatured by the presence of high acid itself. And remember what denaturation means. When a protein is denatured, you don't break the peptide bonds between neighboring amino acids. You simply unfold it from its three-dimensional conformation. And when you unfold a protein, you make accessible to digestive enzymes that might be present internal amino acid peptide linkages that might not otherwise have been available for the digestive enzymes to act on. So in this way, it can facilitate and enhance digestion that goes on in the stomach. Markedly increases the rate of pepsinogen activation, like we just talked about. Pepsinogen is going to be secreted. The same sorts of stimuli that turn on acid production are going to stimulate pepsinogen secretion from the chief cells. But that pepsinogen is inactive. It needs to undergo this cleavage reaction. And that cleavage reaction is stimulated by very low pH, high acid environments. Creates the optimal pH. Once you make that pepsin, it needs an acidic pH to have maximal activity. And the same with the acidic lipases, which would be lingual and gastric lipases. They need to have a very acidic pH to exhibit maximal activity. So it creates the enzyme environment and the pH environment that is right for robust digestion of all those things. And lastly, it activates the D cells, we said. And that's really important because you want to have all this maximum amount of acid production going on when there are nutrients in the system to be worked on, but you want to be able to shut it down effectively when there are no longer nutrients moving through the system, because the acid itself can be so injurious. You want it there when you need it and minimally there when you don't need it. Okay, the very last thing I didn't say, and this was also back on that introductory, on that slide that summarized everything, is sometimes you want to modulate pharmacologically, turn down acid secretion. And why would you want to do that? For instance, if somebody has gastric ulcers, if there are open wounds within the stomach, you want to turn down acid production so that it lets those wounds heal. So there are two different things that you can administer. You can administer what we call the PPIs. Those are proton pump inhibitors, and they are directly targeting those hydrogen potassium ATPase pumps that are actually driving the acid production itself at the apical surface. So this is drugs like Prevacid and Prilosec. You can buy them over the counter. They inactivate those pumps. You have to completely remove those pumps and replace them with new pumps before acid secretion will resume. Because of that, they shut down all acid secretion, no matter what the stimulus was that got it going. So they're very highly effective. And they're effective over long periods of time because, like I said, you can't reestablish acid secretion until you synthesize more of them and get them trafficked and inserted properly into the cell. So they're highly effective, but they're very cost prohibitive. And when they first came out, there was so much price difference between those and the next drugs we'll talk about, that they were very infrequently administered unless you had to use them as the first line of defense because they were so expensive. The other drugs that you can use that are used quite often are called HRBs. These are histamine receptor blockers, and they target specifically that H2 type histamine receptor. Remember, histamine is important because it can drive its own direct effects, but it can also give you these additive effects, boosting the indirect effects of gastrin and acetylcholine, and because it signals through a different pathway intracellularly than the other secretagogues, when it's present in high concentrations, it can give you that potentiated effect. And so you really, as long as histamine can do its job, it can give you much more robust acid secretion than when it's not there. Well, HRBs are drugs that target the receptors for the histamine to act, can significantly turn down the production of acid that's related to histamine, but it can't turn down the acid that would be induced by the release of GRP or just simple gastric stretch in the gastric wall, and the presence of, detecting and being turned on, gastrin secretion being turned on just by the presence of particular amino acids and stuff. So it's not able to block all acid production, so it can't shut it down as thoroughly as the PPIs can. But these were often preferred drugs because, at least when the PPIs first came out, they were so much more expensive, and these were cheaper and would often be used as first lines of defense. We said earlier already that the stomach secretes intrinsic factor, and intrinsic factor is a B12 cobalamin binding protein, right? Turns out that the stomach actually makes two types of vitamin B12 binding proteins. It makes the intrinsic factor, and it makes a substance called haptocorrin. And the haptocorrin, I'm not sure exactly where it comes from, what particular cell types. I've never seen that expressed. It might even be from the same cells, but we know for sure that the intrinsic factor comes from the parietal cells. The same sort of secretagogues that stimulate the parietal cell to release acid also stimulate it to release the intrinsic factor, but it's made, differently. It's packaged in different secretory vesicles, and so you have to target those different vesicles, and the process of its actual release is separate from, but kind of parallels that you would see for the release of acid. In the stomach makes both of these B12 binding factors, but in the conformations that they have and at the pH of the stomach environment and stuff, the intrinsic factor does not have a high affinity for binding the cobalamin. When it's released through the acid actions of acid and motility actions in the stomach, the cobalamin is actually taken up and primarily bound by the haptocorn, which has a much higher affinity for it in the stomach. And so that carries and stabilizes the cobalamin while it's in the stomach. After the contents of the stomach move into the small intestine, there's a pH change in the environment. It goes from being highly acidic to being a more near neutral lumen environment. And that changes the conformation of the intrinsic factor and gives it a heightened affinity for the cobalamin. And also, intrinsic factor is resistant to the digestive effects of all the digestive protease enzymes that are present in the lumen of the small intestine. But haptocorn is sensitive to all of those, or at least some of those digestive enzymes that are proteases active in the small intestine. So the haptocorn actually gets broken down by the digestive enzymes and gets removed, and the cobalamin then has an increased affinity for the intrinsic factor, binds it, and because that is resistant to the digestive enzymes, stays bound to the intrinsic factor in an intrinsic factor B12 complex that travels all the way through the small intestine and goes down to the distal ileum. Way down in the distal ileum, and make a special note of this, way down in the distal ileum, most all your other nutrients and fluids are going to be absorbed in the proximal small intestine, so in the duodenal region and in the jejunum. But in the distal ileum, all the other nutrients have primarily been absorbed up in the other earlier parts of the small intestine. The distal ileum is important for absorbing two different things, one of which is vitamin B12. And that is because in this region, there are epithelial cells that have projecting out into the lumen of the small intestine down here in the distal ileum, specific receptors that don't recognize B12 by itself, and they don't recognize intrinsic factor by itself, but what they can recognize is the intrinsic factor B12 complex. And when they see that complex, it binds to those receptors. That drives receptor-mediated endocytosis, and it brings the B12 into the body in that fashion. The other thing that we'll learn when we talk about the intestine is the distal ileum is also a site where we have major reabsorption and uptake of bile acids that have come down through the intestines in bile. The body needs 3 critical micronutrients to make mature functional red blood cells, one of those of which is cobalamin. So it needs iron, and it needs folate, which is vitamin B9, and it needs cobalamin, vitamin B12 that we've been talking about. Anytime you have a decreased oxygen carrying capacity in the blood, we call that an anemia. And anemias can be caused by too little hemoglobin or too few red blood cells or any combination of the two. If you have anemias, they're actually named by what's the underlying cause of the problem, and the problems can either be related to underproduction, because maybe you don't have enough of these key critical micronutrients that you need for their production, or you can have overdestruction. You can have excessive destruction. So you might have sickle cell anemia or something that makes the cells more fragile and more susceptible to breakdown and elimination. So there, like I said, there are different types of anemias, and they're all based on, and they're named according to what their problem is, or they're categorized according to what's the underlying problem. So there's a type of anemia that is called pernicious anemia. And this is an anemia that is due specifically to the insufficient amounts of B12 in the body, specifically related to the fact that you don't have enough intrinsic factor being produced in the gastric mucosa. So that's the underlying cause is a lack of intrinsic factor. Secondary to that, then you don't get the B12 absorbed into the body appropriately. You usually lack the intrinsic factor because you've had an autoimmune destruction of the parietal cells that are responsible for its production. When you do have a pernicious anemia, your red blood cells oftentimes will be very macrocytic. It's called megaloblastic. When you have megaloblastic cells, they are very much larger size than normal cells, so the red blood cells themselves are megaloblastic, and you also see a hypersegmentation of white blood cells that are called neutrophils. So these are kind of classic indicators that you would have potentially a vitamin B12 dependent sort of production problem. You can also see megaloblastic cells if there are folate deficiencies as well. So just to reiterate a few things we've said already, what kind of carbohydrate digesting enzymes are going to be present in the stomach? The stomach's not going to add any new ones, and we know already from earlier in the discussion, the amylases that came down through the salivary secretions are only going to work temporarily, then they're going to be denatured, and there's going to be a temporary pause in carbohydrate digestion. We talked about the fact we're going to get the pepsinogen, the inactive precursor, the zymogen. That term zymogen is an inactive precursor. So a word that suffix has inogen on the end means whatever I'm talking about, I'm inactive until you modify me and make me biologically active. So we know those chief cells can secrete the pepsin. When pepsin is made, pepsin is going to be an endopeptidase. We'll talk about how that works in just a minute. We said we're going to get gastric lipases from those chief cells up in the fundus part of the body of the stomach. And this is going to start to work specifically on the triglyceride components of our lipids that we've digested. So this is going to be the enzymatic modification. So just mention a few more things about the pepsinogen secretion and activation. We know it comes from the chief cells and some of the next cells. We know it's an inactive precursor called a zymogen, and it needs to be modified to gain biological activity. It has to undergo that catalytic reaction that cleaves some of the amino acids from its N-terminus. That influences how the rest of the molecule folds up. When it refolds, it actually creates an active site that can then do protein digestion, can cleave peptide linkages that it couldn't cleave before. So this reaction, as soon as pepsinogen is formed, it can spontaneously go through this reaction, but it goes through it spontaneously at a relatively slow rate. This reaction, however, is very pH sensitive, and it's markedly upregulated when the pH drops below 3.0. And that's because now it has gone from being in a pH region that might not have been optimal for pepsin activity to a pH optimum that is very optimal for pepsin. So the pepsin that has already been generated then can start participating in driving more and more very rapid cleavage of pepsinogen that's present to pepsin. So when the pepsin is already there present and active to some degree, then any more pepsinogen that's released is almost immediately converted to pepsin as soon as it's released by those chief cells. The secretagogues that would activate those chief cells and get them to increase their release of the pepsinogen would be, again, acetylcholine acting through M3 receptors and increasing cyclic AMP, and hydrogen present itself, because the hydrogen, the acid, when it's present, it activates these short loop E and S pathways that stimulate the chief cells to release pepsinogen. And secretin, we're going to learn secretin is activated when acid is present down more distally in the intestines. It's going to activate S cells. The acid that's coming in from the stomach will activate S cells to make secretin. And secretin also acts through a cyclic AMP dependent pathway, and so it can alter cyclic AMP levels in the cell, and those are other mechanisms for turning on these secretions. So these are the big things that are going to get our chief cells going and get our pepsins secreted. So let's summarize then what pepsin does in terms of GI function in the stomach. Acts luminally. That means it's not attached as a membrane-bound protein to the surface of the epithelial cells lining the stomach, but it's released and it's free-floating and it's finding the substrate proteins it's going to act on out in the lumen of the stomach. It's beginning that digestion of proteins in the stomach. It is an endopeptidase, which means that it can only cleave internal peptide linkages. If each of the boxes here is an amino acid and they're all linked by peptide linkages, endopeptidases can only cleave internal peptide linkages. They cannot cleave terminal peptide linkages. So they couldn't release a free amino acid by cleaving the N-terminus peptide linkage, and they couldn't release a free amino acid by cleaving the carboxy terminus peptide linkage. They're not going to make many absorbable end products. The end products of protein digestion are going to be either individual amino acids or di and tripeptides. Well, and they don't make many of these. The pepsins in the stomach account for only 10 to 20% of the total protein digestion of ingested proteins in a meal, in a mixed meal. But their actions are still important, and they're important because one of the things that they do is by cleaving these internal linkages and making shorter and shorter peptides, they're able to expose amino acids that weren't previously exposed. And those can either be detected by the protein detecting sensory components in the G cells to turn on more gastrin production in the stomach and in the small intestine, or they can be recognized by the I cells in the small intestine and be a stimulant for turning on CCK production in the small intestine. What about the digestion of lipids that's going to go on? We know that we're going to have those fundus chief cells also producing gastric lipase. Gastric lipase, like lingual lipase, is an acidic lipase. It's going to be very active at the acidic pH environments of the stomach. So what's it going to work on? Well, first of all, you should understand that over 90% of the lipids that we ingest in the diet are in the form of triglycerides. And if you can't remember what that is, it's the 3-carbon glycerol alcohol, where you have fatty acids esterified through ester linkages to each of the 3 carbons in the glycerol backbone. So this is a triglyceride. This accounts for almost all of the lipids that we consume in the diet. And what kind of enzymes act on those? Lipases. So the acidic lipases, like the gastric lipase and any lingual lipase that is present in species with lingual lipases, they selectively cleave at the SN3 carbon in these triacylglycerols. That's the location where if there are going to be short and medium chain fatty acids attached, they are usually attached at that position. And there are a number of those in breast milk and stuff like that. So that's why the lingual lipases are important for helping digest these. And it begins the partial digestion of these triglycerides in the stomach. But it preferentially cleaves only at this one ester linkage. So it's going to give you mostly a single free fatty acid that gets released and a 1,2-diacylglyceride. This is called a partial glyceride. These are not absorbable because the fact that they have these 2 fatty acid side chains and a very lipophilic region up here, they can't get through the unstirred water layer, that mucus gel layer that is coating the stomach. So they're not really absorbable in the stomach, and they're not going to be taken up there, but they'll pass on into the small intestine and complete their digestion there. In the small intestine, what's going to happen is we are going to have the pancreas contribute another lipase called pancreatic lipase and a coenzyme that works with it called colipase, which needs to be present along with the lipase for it to have significant enzyme action. This particular lipase is not an acidic lipase. It works at a more near neutral pH, and it can cleave with equal ease at either this ester linkage or this one. So it will take the partially digested 1,2-diacylglycerides made in the stomach, and it will cleave off this amino acid and convert them into 2 free fatty acids and the single 2-monoglyceride. It can also take intact triglycerides and cleave at both of these linkages, and again still produce the same end product, 2 free fatty acids and a 2-monoglyceride. And those products are the absorbable end products of triglyceride digestion. So again, it's going to be chief cells in the fundus that are particularly important in leading to this sort of digestive enzymatic activity in the stomach. We have all these enzymes and all the acid being produced. We said that is very deleterious to the surface of the surface of the gastric epithelial lining. And so we have these surface epithelial cells, like we said, and the mucous neck cells that become very important in secreting mucus. They secrete the mucus, they secrete it, the neck cells secrete it from down deep in these glands. It is jettisoned, it's being secreted out under pressure in these very thin, narrow glands. And so it forms these little finger-like trails that jettison and push it up very forcefully in teeny tiny compact columns up through that gel layer and release it out into the lumen. And the mucus, or excuse me, the surface epithelial cells up here are also very important secretors of the bicarbonate that we talked about. So they will actually take bicarbonate out of the bloodstream, remove it from the blood, from the interstitial fluids, bring it into the cell using sodium bicarb co-transporters. So they're using basolateral sodium potassium pumps to create the sodium gradient, and that's pulling in a lot of bicarb from the blood. And then on their apical membranes, they have the chloride bicarb exchangers that we have seen before, and they're using these exchangers to take that accumulated bicarb and put it out into the mucus gel layer that's there. So the mucus ends up trapping the bicarb and highly alkalinizing it. The significance of that is that you form this mucus gel layer that is hydrating and serving as a physical protective barrier overlying the epithelial cells directly. So if this acid that has been, the parietal cells are secreting the acid and it's coming out in the same way, jettisoned up these little teeny tiny compact columns by the pressure gradient under which it's being secreted into the lumen. If that acid starts to come back and make its way towards the surface epithelium, as the acid comes back through this highly alkalinized gel layer, it gets neutralized by the base that is there. And so it's no longer as damaging to the cells. Also because the enzymes that were active out here, particularly the peptidases and the lipases, and they were active at acidic pHs, as they would move down through this gelatinous layer, they would also enter into regions where the pH was very suboptimal for them to have much detrimental effect on the cells. So by having this highly effective alkalinized gel layer, this is one of the big ways that the stomach protects itself from autodigestive effects. And remember, we've already mentioned what the prostaglandins do, that subclass of PGE2s. We've talked about the mechanism by which they decrease acid secretion. We also know that they're going to stimulate now these mucus neck cells and the surface epithelial cells for the production of the mucus and the bicarb. And then if the tissues do get damaged, like we said, they're going to produce potent vasodilators that will bring increased blood to the region to bring the nutrients that you're going to need to rebuild and replace the damaged tissues with new tissues, and also to bring the white blood cells that you might need to eliminate pathogens or other noxious agents that could be causing the tissue damage in the first place. The stomach then has all of these great protective mechanisms to protect itself from the injurious effects that its own acids and digestive enzymes can induce. Even so, there are still times and situations in which all those protective mechanisms are overwhelmed and the acids and or the enzymes do damage to the tissues. And under those situations, we get open sores in the epithelium that are called ulcerations. And if those ulcerations are localized to the stomach lining or to the mucosal lining in the upper small intestine in the duodenum, we refer to them collectively as peptic ulcers. There are two major causes for peptic ulcers. One common cause is the chronic use of NSAIDs. And you'll remember these are the nonsteroidal anti-inflammatories. If these non-specific type anti-inflammatory drugs are used, they inhibit the COX enzymes that lead to PGE2 and other prostaglandin productions. But especially loss of those PGE2 prostaglandins makes the tissues much more susceptible to these sorts of injuries and insults and much less able to protect themselves and repair themselves once those damaging effects occur. In the absence of chronic NSAID use, as you might have in a patient with rheumatoid arthritis or osteoarthritis, where they're taking them all the time for pain and inflammatory blockers, the most other single common cause is infection with Helicobacter pylori. Now, we've talked about how the acids in the stomach kill off and eliminate many of the bacteria that are in the stomach. And why is it then that these particular bacteria could colonize and cause all kinds of ulcerative problems in the stomach? Well, let's look at the bacteria a little bit. First of all, there are some unique things about the bacteria. They make and express on their outer cell surface, on their outer cell wall, an enzyme that is called urease. And urease can catalyze the reaction that combines urea with water to make ammonia and carbon dioxide. And the ammonia that is produced by these actions can serve as a base and accept hydrogen ions or protons, so can neutralize some of the acid in the environment immediately around the bacteria. And that creates a little halo right around the bacteria where it's not exposed to all of that acid, where it's neutralized the acid, and that prevents the acid from killing it off. Second big important thing is these are flagellated bacteria, and so they can burrow right through that gelatinous mucus layer that's overlying the cells and penetrate right down to the epithelial cell layer. Another big thing is when they get there, they produce a bunch of molecules called adhesin proteins, and these adhesin proteins help them to anchor themselves firmly to the epithelial cell surface. And why is that important? Because when they get there and they can anchor themselves, then even all those motor activities like the MMC contractions that powerfully are supposed to be sweeping out the stomach and could clean out bacteria theoretically, don't sweep them away because they're anchored firmly to the epithelium. Then lastly, they can produce a bunch of toxins that can start to break down the junctions that hold cells together and impair cell integrity and cause damage and destruction to these cells. And so they can help lead to the ulcer formation. And then if the ulcers get deep enough and they go down in and they start damaging and the inflammatory responses make more permeable the blood vessels and stuff, then all the nutrients and the oxygen and stuff that's being delivered theoretically to the cells by the bloodstream is then made also available to these bacteria, and then they really thrive. And so they take off and they colonize these sites and they keep them from healing and they let them spread and they make these conditions chronic and sometimes very painful, debilitating situations. So depending upon where you might have H. pylori localizing, you can either have these ulcers restricted primarily to the stomach or to the duodenum. So if you have H. pylori infection, depending upon where your bacteria are localized and where tissue damage occurs in the stomach, you can either end up with hyperchlorhydric or hypochlorhydric conditions. So hyperchlorhydria is where you have excess secretion of HCl in the stomach. Hypochlorhydria is where you have insufficient or abnormally low amounts of HCl production in the stomach. So let's talk about different locations and localizations of H. pylori and how that can influence whether you have a hyperchlorhydric situation or a hypochlorhydric situation. When you have duodenal ulcers. Duodenal ulcers usually occur when H. pylori infection, if they're due to H. pylori infection, when that H. pylori infection is localized to the antrum down here. Remember, this is the key critical place in the stomach where we have that D cell, G cell paracrine interaction where the D cells, if there's a lot of acid present, the D cells in this region can sense that acid production and they can, in response to that, activate, release somatostatin, and by paracrine interaction, somatostatin can turn down gastrin, and by turning down gastrin can have a marked influence on turning down acid production in the stomach. Well, if you have a lot of H. pylori infection down here, then the tissue damage and the disruption that occurs actually blocks effective D cell breaking of the nearby neighboring G cells. And so since the G cells, you can't effectively put the brake on their gastrin secretion, they secrete a lot more gastrin than normal. So you have hypergastrinemia, too much gastrin in the blood. And because you have these cells up in the oxyntic gland still intact, your parietal cells are intact and functional, and they respond to that increased gastrin in the bloodstream with increased acid production. Then you end up delivering too much acid to the duodenum, you overwhelm its capacity to neutralize all that acid, and then you get the ulcerative damage occurring down here in the duodenum. If, on the other hand, you had your, well, before I go there, let me say that there's one other condition where we could also have hypergastrinemia, too much gastrin present in the bloodstream that is driving too much acid production up in the oxyntic glands in the stomach. And this is the condition of Zollinger-Ellison syndrome. In this syndrome, this is where you have a gastrin-secreting tumor. So you have tumor cells that are abnormal tumor cells, cancerous cells. They're secreting gastrin, and it's not under the normal control of this typical G cell, D cell negative feedback loop that is so effective at controlling acid production under normal circumstances in the body. So you get all this excess gastrin in the bloodstream, drives all this excess acid production up here, delivers all this excess acid down into the duodenum, and again, you would get excessively high acid here that overwhelms the neutralizing capabilities in the duodenum, and you get your ulcers there. Let's contrast that with what if we had H. pylori infection, but instead of having it localized down here, we had it throughout the stomach. It was just kind of generalized and present throughout the stomach. When that's the case, you end up usually having a hypochlorhydric situation. And why do you have a hypo, an under secretion of the hydrochloric acid in the stomach? Because much of your tissue damage is occurring up in the oxyntic glands, and it is actually leading to the damage, destruction, and replacement sometimes of the tissues up there with scar tissue. And so you're simply destroying your cell capacity for acid production. So you end up not having enough acid being produced in the stomach. This can predispose to bacterial overload in the stomach, because bacteria that come in on the foods and stuff you eat isn't sufficiently neutralized and killed off by surface sterilization. And because you have chronic insults going on over time in the stomach in individuals with untreated or poorly treated H. pylori infection that is throughout the stomach, this can predispose repeated bouts and episodes of ulcers and healing and new ulcers developing, and all of that can be a predisposing factor that gives you increased risk of developing gastric cancers and lymphomas. So it's very important, if you do have these conditions going on, to get them assessed and treated. And I didn't mention it on the previous slide, but when you find out that a patient is ulcer positive, they have peptic ulcer disease and they're H. pylori positive, the first thing and most important thing to do is to put them on PPIs or HBRs, histamine blocker, HRBs, histamine receptor blockers, to turn down the acid production. So it stops doing all of its injurious effects to the tissues and gives them time to heal. But then the second thing you want to do is treat these individuals with broad spectrum antibiotics that will kill off the H. pylori infection, and also hopefully minimize bacterial load that's being delivered further on in the GI tract. So very important to eliminate the H. pylori infection, and oftentimes in these individuals, if you do get rid of and eliminate the H. pylori infection, the ulcers will not recur. So very important to treat these conditions effectively. That then pretty much wraps up our discussion of what goes on in the stomach. How does the stomach do its job? What are its jobs? How do the secretory and motor activities facilitate the digestion that goes on there? And a little bit about what minimal absorption goes on there. We know that the chyme is going to next be delivered to the small intestine, and that's the big player. That's where all macronutrient digestion is going to complete, and where all macronutrient, most micronutrient, and most fluid absorption is going to occur. So it's really the big player in the entire system. But the small intestine can't do its job properly without the contributions that come to it from the hepatobiliary system, which is the liver and the gallbladder and the duct system that produce bile, store and concentrate bile between meals, and then deliver bile into the duodenum part of the small intestine. And also the exocrine pancreas, which is going to make a lot of high volume, highly alkalinized fluids that are rich in digestive enzymes that it too is going to dump into this first part of the small intestine, the duodenum. And so we have what we call this duodenal cluster unit where all these three things merge and come together. And so as we move into, we're going to first talk about the contributions and the functions and actions of the exocrine pancreas. And then we'll talk about the hepatobiliary contributions. Then we'll move into and talk about what goes on in the intestines and wrap up the lecture series. So while we have this slide, I want to just point out a few things and make sure we're all clear on terminology going forward. The hepatobiliary system is, like I said, the liver that makes bile, the gallbladder that stores, concentrates, and retains the bile in the interdigestive between meal period. And then this complex duct system that is going to deliver that bile down into the small intestine. So this duct system is comprised of the right and the left hepatic ducts, which drain bile from the liver directly. As soon as they leave the liver, they merge into a common duct that we call the common hepatic duct. That common hepatic duct then bifurcates and gives rise to the cystic duct that can take bile into and out of the gallbladder, or continues on in what we call the common bile duct. The common bile duct comes down. We will see that it is going to merge with the main duct that is draining the exocrine pancreas secretions right before both of those ducts move into the small intestine. So if you opened up the small intestine and you looked at the point at which they enter, you'd see a little bump on the wall around this point of entry. That's called the ampulla of Vater. And right around this point of entry is a band of smooth muscle that we've talked about before. That's the sphincter of Oddi. And the tone of that sphincter of Oddi is going to determine if these hepatobiliary and pancreatic exocrine pancreas secretions can get into the small intestine or not. Once they get into the small intestine, then all of the secretions that are added are going to help the small intestine to do its job. So with that understanding in mind, we're going to go in and finish the rest of today's lecture talking about the exocrine pancreas. So remember that an exocrine gland is a gland that secretes whatever secretory products it makes through a duct system to an external body surface where those secretions are going to act externally. So the pancreas is going to secrete its secretory products through a very complex duct system that is very similar to the secretory units that we talked about in the salivary glands. So we're going to have these blind-ended pouches with acinar cells that make a primary secretion. They pass it through a very complex duct system. Some of the duct cells are going to modify the primary secretions and then deliver them into the small intestine. And remember, the small intestine is part of that tube that just happens to be running through the middle of your body, but it's continuous with the external environment. So essentially, it's putting these secretions on the outside of your body, and they're going to act there in that tube, which is technically outside your body, just in the tube passing through the center of your body, right? And so what is the main job of the exocrine pancreas relative to digestive system function? This is it, to deliver a high volume, highly alkalinized fluid, very rich in digestive enzymes or enzyme precursor zymogens into the duodenum. That's its big job. If you go back, like I said, the secretory process is very, very similar to what we saw in the salivary glands. So go back and review your lecture notes on salivary secretions to remind yourself how that process occurred. In the exocrine pancreas, it's going to occur almost identically with a couple of major exceptions. What are those exceptions? Remember that in the case of salivary glands, that was completely under the neural control of the autonomic nervous system. In the case of the exocrine glands, the control is going to be neurohumoral. We're still going to have neural networks that are in part regulating some of the process, but we're also going to have the secretory process influenced a lot by hormones, and specifically the hormones cholecystokinin and secretin, which are coming from the small intestine. Second big thing is that remember in the duct cells, which were responsible for modifying the primary secretions, remember in the salivary glands, your acinar cells made a primary secretion that was very plasma-like and it was isotonic. But as it moved through the duct cells had very tight cell-to-cell junctions. They reabsorbed lots of ions and not much fluid because those junctions were so tight they wouldn't let water follow the ions that were being reabsorbed. So you kept a lot of the water, you got rid of a lot of the ions, and you converted that originally isotonic primary secretion to a hypotonic end secretion that made it into the oral cavity in the salivary glands. Down in the pancreas, the situation is a little bit different. The cell-to-cell ducts in those ducts that are modifying the primary secretions are much, much looser, and they will let water follow the ions. So the primary ducts or the primary acinar cells make a primary solution. It's going to be a solution that has all of these digestive enzymes or precursors in it. It's not going to have a lot of volume. It's going to have a minimal amount of volume. It's not going to be enough volume to really flush those digestive enzymes through the whole duct system and out into the small intestine. And it's not going to be highly alkalinized. But the ducts are really loose, and so as the ducts modify it, they're going to add a huge amount of base to it, of bicarbonate alkali base to it, and the base that they add is going to create an osmotic gradient, and because water can follow the ions, the more bicarb you dump out, the more water is going to move into the ducts. That's going to give you the high volume, highly alkalinized fluid that will dilute out all those digestive enzymes and carry them effectively into the small intestine. CCK is going to be very, very important in regulating the exocrine pancreas and coordinating its activities with the small intestine. So what is it? Where does it come from? How does it exert its influence? CCK is a peptide type hormone. It is made from a particular population of cells that are present in the epithelial lining of the small intestine called I cells. So these I cells are going to be present, and they are continuously monitoring and looking at what's coming down through the lumen in the environment where they're at. And there are going to be 4 major stimuli that activate these I cells to release their CCK. Products of fat and protein digestion that went on up here in the stomach. So intact proteins aren't as effective at activating those I cells, but those peptones, remember the partially digested proteins that were made by the actions of pepsin up in the stomach, are very effective at activating the I cells. Again, intact triglycerides that are up in the lumen of the stomach, not particularly effective in stimulating these I cells, but the partially digested products of those fatty acids that were partially digested up here. So especially the free fatty acids that were released by the actions of the gastric lipases are very important in stimulating these I cells. There are also some CCK releasing peptides, just peptides that are made by the epithelial cells in the small intestine itself, that can influence and activate these I cells to secrete CCK. And lastly, there's going to be a particular protein that comes from the acinar cells, a secreted product that comes from the exocrine pancreas into the small intestine, and this is called monitor peptide. So monitor peptide is going to come in along with the acinar cell secretions, and when it gets into the small intestine, it's going to be the 4th substance that will stimulate the I cells to make CCK. So that's what CCK is, and I didn't mention, but especially important amino acids in stimulating the activity of those I cells are going to be phenylalanine, valine, methionine, glycine. When we get CCK released, what does CCK do? CCK acts very locally. It gets released into the interstitial fluids bathing these cells, and it actually happens to bind CCK receptors. And these are going to be CCK1 or CCK-A type receptors, which are closely related to the CCK-B receptors that we said were the ones that mediated gastrin effects. But the CCK-A receptors have a much, much higher affinity for CCK than they do gastrin. So they mediate the CCK effects. CCK is going to bind binding sites on those sensory afferent neurons, that's going to activate vagovagal reflexes. So an afferent signal goes into the CNS, carries information in, says, hey, CCK is telling us that there's some luminal contents here that need to be digested and need your help. And then efferent signaling is going to come back, but not go to the intestine, this time come to the exocrine pancreas. It's going to synapse with and use cholinergic postganglionic fibers, and the acetylcholine that they release is going to be the main stimulant for these acinar cells. So these are going to be the cells that release all the digestive enzymes and the monitor peptide in minimal amounts of fluid into this blind-ended acinar pouch. So the CCK, once it gets released, drives those vagovagal loops that release acetylcholine, stimulate, target specifically the acinar cells, and that's where they exert their primary effect. And the effect is to give you all those digestive enzymes. The little dotted line here says that CCK can exert a secondary effect by itself. This dotted line means it's not able to give you much of a robust response in the duct cells that line the ducts and modify the primary secretions. But if it is present at the same time that secretin is present, secretin is going to be the hormone that primarily influences these duct cells. And if acetylcholine is present in small quantities at the same time secretin is there, it can give you a potentiated effect. And secretin's effect is to drive a whole bunch of bicarb secretion out into the fluid, so to highly alkalinize these fluids in response to the presence of acid that it saw coming down in through the system. So its job is to give you a bunch of bicarb out here. The bicarb builds up, creates an osmotic gradient, pulls a bunch of fluid in, gives you a huge amount of alkalinized fluid. That's enough to solubilize, dilute, and sweep all these digestive enzymes into the small intestine. So what is a potentiated effect? Again, I just want to remind you that if you were measuring bicarb as an indicator of the pancreas response to secretin, if you gave secretin alone, secretin would give you a modest but sustained, significant and sustained increase in bicarb secretion in the lumen there. If you gave phenylalanine, phenylalanine is one of those amino acids that would be a very potent stimulant to give you endogenous CCK production. And if you gave phenylalanine and it gave you endogenous CCK, the CCK would give you a much weaker and inconsistent response by those duct cells that secretin would target. If they had an additive effect, then you would expect that if you gave the two of them together, you might get some sort of an effect in about this range, right? But that's not what happens. If you give the secretin in conjunction with a small similar amount of phenylalanine that you gave down here, you get a much, much higher response than what the expected additive effect would be. And that heightened response is what we call that exaggerated or potentiated synergistic effect. And so it helps to give this synergistic boost or potentiation to secretin action. And the last thing it's going to do in the pancreas is it's going to help to relax this sphincter of Oddi right at the end where the pancreatic duct and the hepatobiliary duct merge and come into the small intestine. So importantly, it's going to relax the sphincter of Oddi, and that decreases the tone and decreases the resistance and allows the pancreatic and the hepatobiliary secretions to enter into the duodenum. There are also actions of CCK outside the pancreas. So what else does it do? We've talked before about it has introgastron effects. It's one of those numerous substances made in the small intestine that feeds back and constricts the pyloric sphincter. So this time increases the tone of the pyloric sphincter. It relaxed the sphincter of Oddi, but it increases contraction and tone in the pyloric sphincter. So it makes that sphincter have a really big, tight sieving effect. It slows gastroduodenal transit and the emptying of stomach contents into the intestine, and also slows down acid production up there. It goes all the way up to the hypothalamus, to where your feeding center is, in the center that knows if you're sated and full or if you still need to eat. And it induces a feeling of satiety. So it turns down appetite. and drive for increased food intake up in the hypothalamus. That's why if you're going out to a meal and you're trying to lose weight, you know how at a typical meal in a fancy restaurant, they'll bring you a drink first, and then they'll bring you a salad, they'll bring you all this bread and all these things that are really, really high in carbohydrates and could really pack the pounds on you and put the pounds on you. And then they bring you your steak or fish or, you know, whatever your main protein part of the meal after the fact. If you ate the protein part of the meal at the very beginning, before they brought you the wine and all of the salad and all of the breads and the dessert list after that, you'd rapidly start secreting CCK, and CCK would go up to your brain and say, hey, I think you feel sated. And you would end that meal earlier. You might not drink the whole glass of wine. You might pass a little bit on your salad. You might not even eat any of the bread. You might completely blow off the dessert list because you just didn't have that much more of an increased appetite. It acts over in the gallbladder, and it increases the contractile activity in the wall of the gallbladder. So it causes the gallbladder wall to contract around its hollow tubed lumen. That increases the intraluminal pressure in the gallbladder and helps drive out any of the accumulated concentrated bile that had accumulated there in the in-between meal state. And lastly, it can exert trophic effects on the pancreas itself. And so trophic effects are simply, it stimulates transcription, translation, and protein production in cells, drives cell division, and so maintains the number of cells and the activity of the cells that are present in the. Let's look for a minute at what kind of enzymes the exocrine pancreas via those acinar cells actually contribute into the duodenum to help finish up nutrient digestion that needs to go on there. It gives you enzymes that can work on any particular type of macronutrient that could be consumed in a mixed meal sort of a situation. Gives you a number of enzymes that can work on proteins. Many of these, we've learned that the suffix inogen means I'm made as a zymogen, and I am inactive the way I'm made. I have no biological activity, but if you modify me, I can do something. So all of these enzymes that their name ends in inogen, they're made as zymogens, those inactive precursors that need to be activated somehow. Similarly, if their name actually begins with the prefix pro, that also indicates that they are made in zymogen form and they need to be modified. Many of these enzymes, when they are modified and they are converted to their active enzymes, all of these top four act as endopeptidases, similar to what pepsin did. They cleave internal peptide linkages to cut bigger proteins into smaller and smaller proteins and peptides. The carboxypeptidases are enzymes that can cleave, they are exopeptidases. They can cleave terminal linkages at the carboxy terminus end of the protein. So the carboxypeptidase A members actually will cleave if that final amino acid is an acidic acid. Amino acid, they'll cleave and release acidic acid amino acids from the carboxy terminus. And those that belong to the carboxypeptidase B family will cleave peptide linkages and remove basic sort of amino acids that end up being in the terminal position at the carboxy end of polypeptide chains. So it turns out that all of the protein digesting enzymes that it makes them all in zymogen form. But it makes, not all zymogens that it makes are proteases and work on proteins. For instance, it makes phospholipase A2, and it makes it as a pro-phospholipase A2 zymogen that has to undergo cleavage and become this active phospholipase A2. This is an enzyme important not in working on triglycerides, but more specifically in removing the fatty acids off of phospholipid molecules. It makes a lipase, well, it makes a number of its enzymes that it makes as active enzymes, that they're not made as zymogens that need to be converted. They're active enzymes if you gave them the right pH environment, and if they happen to need cofactors and you had the right cofactors available and active for them to work with, they could do their job without any modification. So it's going to contribute alpha amylase, just like the alpha amylase that came from the salivary glands. It's going to make more of that, dump it in. So remember that the alpha amylase was going to be inactivated in the stomach before it could completely convert all your starches and glycogens to maltose, maltotriose, and alpha limit dextrins. So you'll have about 30% of those that never got modified. The exocrine pancreas is going to give the small intestine a bunch more of those and say, use these to convert all of those as yet unmodified starches and glycogens into their end products. It's going to give you a pancreatic type lipase. This is not an acidic lipase like the lingual and the gastric lipases were. This is a lipase that works at a neutral, near neutral pH, like you'll see in the lumina of the duodenum in the small intestine. Needs a cofactor to have much activity, so it needs to have colipase with it. The exocrine pancreas also makes that cofactor, but it makes it in an inactive zymogen form. So it has the enzyme. The enzyme won't have much activity if it doesn't have its cofactor, and it makes the cofactor in a form that isn't readily able to act in the pancreas itself. It makes this carboxyl ester lipase. This is a broad spectrum enzyme that can cleave lots of ester linkages, so it can cleave at any of the positions in the triglycerides and remove fatty acids from any of those. It can cleave fatty acids at any of the positions in your phospholipids. It can cleave fatty acids off of esterified cholesterol, which is kind of where it gets its name. It can cleave fat-soluble vitamins that happen to be esterified and linked to other nutrients in that way. It's going to give you RNases and DNases in sufficient quantities that they could work on any nucleic acids like RNA and DNA molecules that you might have ingested. So it gives you this whole broad spectrum of enzymes that can do anything, break down any of these types of macronutrients. So it's providing enzymes that actually have the capacity to chew the pancreas itself up. So how does it protect itself and minimize the possibility of autodigestion by all these digestive enzymes that it makes? Well, first of all, it makes many, many, many of them in zymogen form. And then importantly, it does not activate those zymogens until after they leave the pancreas under normal circumstances. We'll learn, I have this one starred because this one is, of all of the zymogens that are produced, this is the most potentially dangerous to the pancreas, because if this gets activated prematurely, the trypsin that it produces then serves as the enzyme that cleaves all of these other zymogens that are made in the pancreas into their active enzyme forms. So if the trypsinogen would prematurely activate within the pancreas, it would start this cascade of activating all of these other enzymes, and that would certainly overwhelm the pancreas's mechanisms to prevent autodigestion, and it would cause damage and a lot of destruction within the organ itself. So because of that, this one is so potentially dangerous to the pancreas. In all the little secretory granules that have trypsinogen in it, they release in 1-to-1 molar ratio another molecule called trypsin inhibitor, so that if the trypsinogen prematurely activates in the pancreas, you'll have in 1-to-1 molar ratio an inhibitor that's right there with it that hopefully will inactivate it right there locally before it can start this cascade of events that would activate all the other zymogens. The one thing that I put this slide on for, it says a lot of what we said in the previous slide, shows you that for all of these protein digesting enzymes that come from the exocrine pancreas, they're all made in the inogen or the pro-hormone zymogen form. So they all need to be activated. And importantly, the activation normally takes place outside of the pancreas. So we wait till we get everything into the small intestine, and that's where you have a brush border enzyme called enterokinase. This enterokinase is an integral membrane protein that is present in the apical membranes of the enterocyte cells, the endothelial cells that line the small intestine. And its active site is looking out into the lumen, and it's specifically looking for this substrate because it binds trypsinogen, causes it to undergo a cleavage reaction that converts trypsinogen into trypsin. And we said the importance of that is that once the trypsin is formed, that is the enzyme that will convert all of these other inactive zymogens to their active forms. So this just is reiterating that point. Second thing, why do we have so many darn different types of protein digesting enzymes? Because there are lots of different amino acids, and depending upon how they're put together and what combinations and stuff, it can make it hard for some of these amino acid digesting, peptide digesting enzymes to get in and cleave particular linkages. So each one of these has a different sort of site along the length of a polypeptide chain where it likes to cut, has different specificities for where it likes to cut. I don't want you to know all of these. The only point that I want to bring out and emphasize again here is that I said at the beginning of the lectures when we talked about having high proline content in proteins or high glycine content, the incorporation of large amounts of proline or glycine residues in proteins within the GI tract makes those molecules very, very stable. And it's very hard for a lot of our digestive enzymes in the human digestive tract to cut right at a proline, right at the peptide linkage that is attaching a proline to another amino acid or to the N-terminus end of the protein. So it doesn't like to, like for instance, the carboxypeptidase A and B type carboxypeptidases that will release individual amino acids from the N or the carboxy terminus end of polypeptide chains. Won't cut if that last amino acid is a proline. Won't cut if that last amino acid is a proline. And this one won't even cut if the one right before the last one is a proline and the last one isn't the proline. It still won't cut that last one off if there's a proline immediately preceding it. So the presence of all these prolines makes it hard for our digestive enzymes to get in and do their jobs effectively, causes some steric hindrance there that prevents them from working okay, and that's why makes those sorts of proline-rich and glycine-rich amino acid polypeptide chains in our bodies relatively resistant to the effects of our digestive enzymes. I think this is a really nice place to end today's lecture. We didn't get quite all the way through the exocrine pancreas material I want to cover, but we'll finish that up early in the next lecture. We'll move into and talk about the hepatobiliary system and its contributions as part of that duodenal cluster unit. And then we'll move in and talk about we'll start to talk about what goes on in the small intestine. So that's where we're going for lecture 4. Thank you for all your time today. It's been wonderful working with you.